Good morning, everyone. Welcome to our session. Thank you very much for your support, Dr. İbrahim Sözdutmaz, for us, for the Congress. Now the session's moderator is İbrahim Sözdutmaz. Yes, Professor, your turn and welcome.
Thank you for your kind invitation to me as a moderator. Good morning, everybody. In this session today, it is the last day of the Congress, and in this session there are like 16 presentations in this session. So we are going to start with the first presentation with Belma Dayı. She's going to present her presentation, synovial hemangioma of Stifle joint of Kangal dog. We are listening, Belma.
Thank you so much. I'm sharing my screen. Oturum sahibi katılımcının ekran paylaşımını engelledi diye bir not düşüyor hocam.
Ben bakıyoruz hocam. Bir daha deneyebilir misiniz? Can you try it again, please?
Thank you. Can you see my screen?
We can.
Yeah, your screen is okay. It's going on.
Thank you. Good morning, dear researchers. My name is Belma Dayı. I'm a research assistant at Afyonkarahisar University, Faculty of Veterinary Medicine, in the Department of Pathology. Today I'm talking about a case of synovial hemangioma of the stifle joint in a Kangal dog. Synovial hemangioma is a rare benign neoplasm in humans and animals. This neoplasm can be located on any synovial membrane surface, along tendon sheets, or in the spaces within the joint. As you can see in figure 1, it is the most commonly observed in the knee joints in humans and dogs. The symptoms of this tumor can be recurrent, and it can cause swelling, pain, and lameness depending on factors such as size, location, and changes in neighboring structures. Histologically, vascular channels lined by endothelial cells without features of malignancy and filled with thrombi and erythrocytes are observed, as you can see in figure 2. And also, these channels can be separated by fibrous connective tissue in figure 3. According to the studies, only 4 cases of synovial hemangioma have been described in animals. And this study aims to evaluate a rare case of synovial hemangioma histopathologically. The subject was a 10-year-old male intact Kangal dog, and the mass localized in the left stifle joint region. The owner noticed lameness in its left hind leg for 5 months. There was pain resulting from manipulation of the stifle joint. There were no extension or flexion movements. The radiological examination revealed soft tissue swelling in the left stifle joint region. And it was also reported that the dog had no history of trauma and had respiratory distress. For histopathological examination, tissue samples were fixed in 10% formalin. After routine processing, they were embedded in paraffin. Paraffin blocks were cut at 4 micrometers in thickness. They were stained with hematoxylin, and slides were evaluated with a light microscope. For immunohistochemical stainings were performed using a commercial staining kit according to the manufacturer's instructions. Tissue samples were processed routinely, and then sections were subjected to straight buffer in a microwave at 600 watt for 2 times 5 minutes for antigen retrieval. Sections were incubated with primary antibodies against vimentin, actin, and CD31 overnight at 4 Celsius degrees. And when we look at the table 1, you can see the dilutions and primary antibodies. We used 1 to 500 dilutions for vimentin and actin, and 1 to 20 dilutions for CD31. And then slides were incubated with secondary antibody for 30 minutes at room temperature. 3,3-deaminobenzidine was used as a chromogen, and then slides were evaluated with Olympus stream image analysis. When we look at the gross examination, the mass was found to be 1.2 by 0.8 by 1 centimeters in size, dark brown, red in color, and medium hard. Histopathological examination. As you can see in figure 6 and 7, arrows indicated the vascular channels filled with erythrocytes and lined with well-differentiated endothelial cells, and stars indicated the significant proliferation in synoviocytes. And as you can see, large blood containing areas found around the synovial cells. When we look at the immunohistochemical stainings, in figure 8, arrows indicated the CD31 expression in endothelial cells of vascular channels. In figure 9, as you can see, arrows indicated the actin expression in smooth muscle cells of vascular channels. And also figure 10, arrows referred to vimentin expression in the stromal connective tissue around the blood vessels. Outcome and follow-up. Unfortunately, it was reported that the dog died in a private clinic, where it was treated approximately 1 month after the diagnosis of synovial hemangioma. It was reported that the dog's continuing respiratory distress and old age could have contributed to its death. Our study is the first to ever evaluate synovial hemangioma in an animal in Turkey. Diagnosis of synovial hemangiomas is usually difficult. In this case, in addition to the routine histopathological methods we use, immunohistochemistry was also helpful in confirming the diagnosis of the synovial hemangioma. It has been reported in human medicine that synovial hemangiomas are sometimes observed as post-traumatic lesions. However, in some cases observed in dogs, there is no history of trauma. It's thought that surgical excision is an effective treatment method in cases of synovial hemangioma, not complicated by other disease, and that its application is important to avoid degenerative changes that may occur, especially in the articular cartilage. This case report is distinguished due to the limited number of similar studies. We hope that it will help in the future to evaluate patients with similar findings. Thank you for your attention.
Dear Belma Dayı, thank you for your nice presentation. Is there any questions about the presentation? Thank you. We are coming to next presentation. Next presentation is will be presented by Aytaç Ünsal Adaca. She will present her study, pros and cons of distance learning in teaching history of veterinary medicine. Yeah, we are listening to you.
Thank you, dear moderator. Dear moderator, Professor Söz Tutmaz, dear researchers and participants, good morning to everyone. Welcome to my presentation. I'm going to share you to my presentation. I hope you can see my screen now. Is it okay?
Yeah.
Yes.
Yeah, that's okay.
Thank you very much. I am from Ankara University, and I'm going to show you my study, which is about pros and cons of distance learning. For introduction, I'm going to show you what is distance learning. It's a little summary for us. Distance learning is also known as distance education, and it is an active learning method for students. In this method, training activities are carried out through online resources. Students and teachers are not physically in the same environment. They are not meet in a classroom. In this study, I am going to show you pros and cons of these courses, they are conducted through the distance learning in this academic period. In these courses, we held online for 1 hour a week for 14 weeks in this year, and we have totally 2,021 students and 1 lecturer, and 1 lecturer is me. I use the distance learning software of Ankara University. You can see my screen now. We use this software for teaching these courses. I separated the results as 6 parts. One is pros of distance learning for the students. In this part, I can say the students do not have to take notes during the lesson. It is the very important pros of this learning. All students can observe the lecturer's screen as like this congress. The biggest pro of this method is students are able to watch records whenever, wherever, and as much as they want. It is the most important part of this section. Students who are shy from asking questions has the chance to communicate with the lecturer in writing. It is the most important, valuable result of this section. And the second section is about cons of distance learning for the students. The biggest cons is lessons were asking question and active participation in the lesson were limited almost to a few students. It is very big cons for us. The third part of the results are about pros of the distance learning for the lecturer. In this part, we can see lecturer is not physically present in the classroom, and lecturer can enter the lesson instantly from anywhere with her own computer. As for the cons of the distance learning for the lecturer we can say the lecturer did not see the faces of any students, and she did not have the opportunity to meet any of them face to face, and this situation negatively affected the motivation of the lecturer. And pros of the distance learning in general, we can see these results. The courses are being recorded. It is very important for this learning statement for the active learning. All the resources can be accessed via research engines. And in the pandemic rules, with this method, all students followed social distancing rule. It is important also. And what is the cons of the distance learning in general? I can say none of the students use microphones or cameras during the lessons. Questions or answers given were shared via chat box only, and they used as short answers. No question or feedback was conveyed to the lecturer. And very big cons of this method is inability to establish effective communication and mutual interaction between students and the lecturer. I separated conclusion as 3 parts. One is about the courses. History of veterinary medicine course is theoretical course by its nature. So teaching of this course was found to be compatible with distance learning method. It is not about the practice lessons. It is about the theoretical lesson, and it was found to be compatible with this method. And the second is the summary of the advantages and disadvantages of this method. in this method, all students and lecturers have the flexibility to study whenever, however, and wherever they want. This seems very advantageous of this method. The biggest disadvantage of distance learning is student and the teacher cannot interact, cannot communicate with themselves. So it is very important for learning system. And the last I want to say, as a consequence, it can be said that the history of veterinary medicine courses taught by distance learning have many advantages and disadvantages for both students and lecturer. And I can see that the same results at the international reports from most of the countries. And maybe in the future, these methods can be integrated to all the curriculum. We can stay and wait the results of the studies from the another countries. And thank you very much for your attention. If you have any questions or feedbacks, I am waiting for you. Thank you very much again.
Dear Dr. Adaja, thank you for your nice presentation. It's showing our some of the problems with the online education system. If you have any questions about the presentation, can you write on the chat box? And after the whole presentations, we are going to answer these questions. I would like to pass the next presentation. The next presentation is about the effect of using litter materials in broiler breeding on performance, carcass yield, antioxidant status, and some litter parameters and coccidiosis. So the presentation will be held up by Şevket Evci. Yes, we are listening to you, Dr. Şevket.
Hello, the chairs, all participants. I respectfully greet everyone. I will share my presentation. Is it okay?
Yes, it's okay.
Again, hello to chairs and all participants. I
Sorry,
respectfully
we are
greet
only seeing the last one. I think you can, if it's possible, you can go on the first one.
Okay. My name is Şevket Evci. I am working as an assistant professor at Delice Vocational School of Kırıkkale University. In this study, the effect of using different litter materials in broiler breeding on performance, carcass yield, antioxidant status, some litter parameters, and coccidiosis oocytes. For profitability in broiler farming, it is important to reduce feed and maintenance costs, chick mortality rates, and increase the quality of the carcass produced. Also, one of the important factors for profitability in poultry business is litter material and management. Because of its quality affect broiler welfare, behavioral characteristics, some oxidative stress parameters, fattening, performance, and carcass quality. The material used as litter should provide a comfortable environment for the chicks by preventing direct contact of the chicks with the ground and providing insulation, and should improve the care conditions by absorbing the moisture in the feces. The increase in moisture and pH value in the litter causes an increase in the bacterial load. As the pH value and moisture level increase in the poultry litter, ammonia emission from the litter increase. The increase in ammonia emissions deteriorates the air quality of the hen house and causes health problems in chicks. Decreased performance and low quality of manure obtained at the end of production due to nitrogen loss. In this study, effects of litter materials containing separately and equal amounts mixture straw and wood dust on performance, carcass yield, antioxidant status, litter moisture, and ammonia levels, presence and development of coccidiosis oocysts in broiler breeding were investigated. The research was carried out at Kırıkkale University Faculty of Veterinary Medicine Poultry Research Unit at the age of 0 days, 120 rows, 308 breed broiler chicks from a commercial firm were brought to the research unit, and their body weights were weighed and divided into groups. Research was composed of 3 main groups with 4 subgroups, and it was disturbed equally so that there were 10 chicks in each subgroup. The study was organized in 3 main groups as wood dust, ground straw, and mix, and each group included 4 subgroups. The research was carried out for 42 days on the ground, feed and water provided as ad libitum. The body weights were determined as the group average every week by weighing the chicks individually. After it was determined as the weekly average body weight, it was calculated difference between it and the previous week, and that the average body weights was determined for 6 weeks. Feed consumption of the groups was determined by calculating the difference between the amounts of feed put in the feeders for a week and feed left in the feeders at the beginning of the next week. Feed conversion ratios were calculated weekly with total feed consumption divided by body weight gain. The MDA level in the plasma was determined by the method of, as a result of the thiabarbiturate acid reactions in the spectrophotometer. Plasma vitamin A and beta-carotene levels were determined by the method of Suzuki and Kato with spectrophotometric measurement. Plasma vitamin C analysis according to Haig and nitric oxide levels were determined spectrophotometrically by the method of Miranda et al. Plasma total antioxidant status levels according to Errol and total oxidant status levels according to Errol was determined. Oxidative stress index values were calculated with total oxidative stress status divided by total antioxidant status. To determine ammonia, nitrogen, and moisture levels as litter parameters, samples taken from 5 different points, 4 corners, and 1 middle of each subgroup compartment were mixed. According to AOAC, ammonia nitrogen levels were obtained by the Kaldal method. Moisture levels were obtained by drying at 105 degrees Celsius for 24 hours in the drying cabinet according to AOAC. From the first day of the study, samples were taken from each subgroup compartment every day, and coccidiosis oocysts were examined under the microscope with the simple flotation method. In this table shows performance values of the research group. Performance values is body weight gain, feed intake, and feed conversion ratio. This table shows the performance values according to the liters. The highest feed consumption, feed conversion ratio, and body weight gain were in the mixed litter group, but there was no statistical difference between all groups in the table, except feed conversion ratio at 3 and 6 weeks. In this table shows carcass yields of the research groups. Carcass yields groups are hot carcass weight before slaughter body weight and carcass yields. Hot carcass weight and pre-slaughter body weight were the highest in the wood dust group. Carcass yield was highest in the straw litter group. But there was no statistical difference between all groups in the table. In this table shows antioxidant status of the research groups. The factors considered here are total oxidant status, total antioxidant status, oxidative stress index, malondialdehyde, and nitric oxide, also vitamin C, beta-carotene, and vitamin A. The amount of nitric oxide was higher in the mixed group compared to wood, dust, and straw. The amount of beta-carotene was higher both mixed group and wood dust compared to straw group, but there was no statistical difference in terms of other factors. In this table shows ammonia levels measured from weekly litter samples of the research groups. The ammonia level increased gradually and was determinant in the high straw group. There was no statistical difference between all groups in the table. And in this table shows moisture levels measured from weekly litter samples of the research group. The moisture level increased gradually and the highest was determinant in the wood dust group at the 5th weeks. There was no statistical difference between all groups in the table. And conclusion, as a result, it has been determined that the use of wheat dust and straw separately or in equal proportions as a leader will similarly affect on performance, but wood dust will positively affect the antioxidant status. In addition, since coccidiosis oocysts were not observed, the effect on their development could not be determined, and similar studies are needed in the future. Thank you for listening.
Dear Dr. Evci, thank you for your nice presentation. If you want to collaborate for further, studies, you can write your email on the chat box, and if there are any questions about to the researcher, you can also write your questions to the chat box.
Okay.
Thank you for your nice presentation. Dear respected participants, I would like to pass the next presentation. Next presentation will be presented by Mohammed Sheraz. He will present his study, genetic analysis of DMP for GLI2 and HSHH genes in the sheep and goat affected in the Aplasia ani with candidate gene approach. Yes, we are listening, Mohammed. Your voice is not coming. Is it possible to share your voice? there is a sound problem.
Mohammed Sheraz, we cannot hear you. No sound. If there is any problem, we can pass the other academician. Can you solve it now or we pass you? No sound yet.
Okay. There is no sound, Mohammed. If you can stop your presentation and we can pass the other one. If you arrange the sound, we can come up to you. No sound is coming, Mohammed. During that time, if you can arrange. Your sound is not coming. No sound is coming. If you can start over your computer or if you can do something, we can come up to you after the next presentation. Okay. Okay, there's a problem with the sound of Mohammed Sheraz. So we are passing the next presentation. I think Abdulrefa Al-Bahi, if you are ready to present your presentation.
Yes, do you hear me?
Yeah, now we are hearing you. Okay, next presentation will be presented by Abdulrefa Al-Bahi, evaluation of land cover status of protected areas using the GIS and remote sensing techniques. Now we are listening to you, Dr. Al-Bahi.
So do you see the presentation?
Now it's okay.
Okay, so hello everyone. Let me first introduce myself. I'm Ilbahi Abdurrafi, a PhD student at the National University of Ireland in Galway and at the Ibn Zawr University of Agadir. I would like also to thank you for giving me this opportunity to participate in this Congress. So today we are going to talk about evaluation of land cover status of protected areas using QIS and remote sensing, more specifically the identification and mapping of the habitat types in the Stifle National Park. To do so, we have organized this talk in the following way, starting by introduction, followed by material and methods, then results and discussion, and lastly, conclusion. Let's start by a brief introduction about Stifle National Park, which is qualified as a biodiversity hotspot area within the Mediterranean basin, located along the Moroccan Atlantic coast of the Stifle region. It is the first coastal national park in Morocco, created to preserve the high diversity of its continental and marine environments. It takes its name from the Souss and Massa rivers, which host two Ramsar sites of international importance. It is considered the heart of the first biosphere reserve in Morocco, known as the Argan-Ray Biosphere Reserve, which hosts the endemic argan tree, Argania espinosa, a species in need of conservation. So far, very little work has been done to study Sustmaster National Park habitat types, and available data on its current very natural and disturbed habitat sites national park. So the region of interest for include both the marines, 5 kilometers wide in average and 65 kilometers long, with a total area of 53,800 hectares. The marine zone is, however, smaller and measures about 3 miles wide, extending from the coastline of the western.
Thank you for your nice presentation. I think the sixth, we are going to.
I'm sorry? Sorry?
Did you finish your presentation?
Yes.
Okay. Thank you for
Thank
your
you.
nice presentation.
So,
There's a problem. Okay.
do you hear
Okay,
me?
now. Okay, that's okay.
Can I continue?
Okay,
You can, but you have, I think, internet connection problem.
Maybe I'm sorry for this. If it's possible, can you complete this?
Yeah, you
That's-
can complete.
You can complete.
So the Sentinel-2 free access data were downloaded from the website of Copernicus Open Access Hub. 10 spectral lengths were extracted and used for this study. Data processing consisted of the extraction of the region of interest, resampling, and conversion to reflectance, which was performed based on the metadata file downloaded with the images, which contained the required information for the conversion of the satellite images from digital numbers to reflectance values. Data processing and classification consisted of the supervised classification approach. Training areas were created manually for each habitat class, identified in the Sentinel-2 images. These training areas were carefully selected and consisted of polygons drawn over visually homogeneous areas of the images. The classification accuracy estimate was carried out using the method of confusion maps, where information collected in the field is compared with the classes assigned by the user to the map. Additionally, the entire data processing was performed using the semi-automatic classification plugin in QGIS. The supervised classification method resulted in the identification of 16 classes of natural and disturbed habitat. The presented map illustrates the distribution of the identified habitat types, describing the current situation of Sustmaster National Park. The first 11 classes represent natural habitats, while the remaining classes are considered as disturbed habitat. The results of the accuracy assessment of the classified habitat map showed that the overall accuracy of the user and producer accuracies exceeded 85% for all classes, while capacity exceeded 0.81. The accuracy assessment analysis showed that the classified habitat map had a strong agreement with reference data. The provided data revealed that 32% of the total surface of the national park was covered by marine surfaces. The remaining 68% was occupied by terrestrial surfaces. The terrestrial natural habitats were evenly distributed within the study area and represented a share of 65% of the total terrestrial area. They were characterized mostly by shrublands, while on the other hand, the supervised classification resulted in identification of 39.5% of the terrestrial surface to be regarded as disturbed lands. The latter was dominated by cleared lands and overgrazed pastures. The presented case study showed that the combination of relatively high resolution remote sensing data, techniques based on geographic information systems, and field data successfully produced up-to-date thematic habitat map of Stifle National Park. This demonstrated their importance for the planning and management of protected areas, and as they allow for cost-effective monitoring of large and diverse areas of the spatial characterization of habitat types. And thank you for your attention, and I'm so sorry for the technical problems.
Dear Dr. Elbahi, thank you for your nice presentation. If there, from the participants, if you want to contact with Dr. Elbahi, if it's possible, you can write your email to chat box and you can directly contact with Dr. Elbahi. And now we are going to next presentation. Next presentation will be presented by Dilek Yalçın, determination of anti-biofilm and anti-corium sensing activities of silver nanoparticles synthesized by green method using algae species.
Thank you very much, dear Professor. I'm sharing. Today, I will present you our study, names of determination of anti-biofilm and anti-corium sensing activities of silver nanoparticles synthesis by green method using algae species. Today, inappropriate use of antibiotics has led to an increase in the number of multi-drug-resistant microorganisms. As a result, the need for successful development of effective therapeutics against bacterial pathogens has emerged. Recently, silver nanoparticles have come to the fore the research for effective and sustainable solutions. Corium sensing is a mechanism that allows bacteria to communicate with one another. The main function of QS is to regulate vital cell processes like virulence factor production and biofilm formation. Corium sequencing disrupts microbial communication. QQ-deriving molecules can reduce or even stop the production of virulence factors entirely, including biofilm formation. Biofilms are film-like complexes that occur on biotic and abiotic surfaces and are sessile bacterial communities enabled in a self-synthesing exopolysaccharide matrix. Bacteria in biofilms show different characteristics from free-living cells, such as different physiology and high resistance to antibiotics, which makes them a chronic and persistent source of infections. Therefore, there is a need to investigate potential anti-QS and anti-biofilm compounds from natural sources to prevent bacterial infections due to increased multi-drug resistance among pathogens. In this study are materials and methods. Two algae species used in this study, Clepsoridium subtile and Osculatoria princeps, were obtained from the culture collection of Ahi Evran University Culture Collection. These algae species were grown in optimal conditions for 3 to 4 weeks. As you can see, this algae species picture. In our previous study, silver nanoparticles were obtained from existing algae by green synthesis. The characterization of the synthesis silver nanoparticles were performed by several techniques: visual, UV visible spectroscopy, Fourier transform infrared spectroscopy, scanning electron microscopy, and energy dispersed spectroscopic analysis. Antiquarium activity of the compound was determined against Chromobacterium violaceum. The violaceum and quantitative evaluation experiments were performed according to MacLean et al. The extracts of Q subtile fresh and dry biomass and O princeps on the fresh biomass, silver nanoparticles extracts were tested for pigment production inhibition. The violaceum quantitative evaluation experiments were performed according to the Kahn et al. The antibiofilm activity of silver nanoparticles were determined by including clinical pathogens, Staphylococcus aureus, Bacillus cereus, P. aeruginosa, and E. coli. Biofilm inhibition assay experiments were performed according to the Scilacci et al. And our results and discussions: the antiquarian properties of silver nanoparticles algae extracts were investigated using C. violacerium strain. All extracts showed colorless colony formation, which is an indicator of antiquarian capacity against C. violacerium strain, and zone diameters were measured in millimeters. Table 1 and Table 2 show antiquarian sensing activities of algae K. subtile fresh and dry and Osculatoria princeps results. The present study was carried out to find the antibiofilm activity of silver nanoparticle extracts of algae against the pathogenic bacteria Staphylococcus, Bacillus cereus, Pseudomonas aeruginosa, and Escherichia coli. The antibiofilm activity of the biosynthesis silver nanoparticles showed a reduction in the biofilm formation by all test pathogens. And here are the Table 3 and Table 4 show antibiofilm activity of the synthesis silver nanoparticles from algae. And the conclusion: in this study, the silver nanoparticles extracts of Q. subtile and O. princeps significantly inhibits the biofilm formation and requires control virulence factors in P. aeruginosa and Staphylococcus. The study revealed that macroalgae are able to act as a biocontrol agent against pathogenic bacteria in aquaculture. And here are our references. And thank you for your interest in our work. Thank you very much.
Dear Yalçın, thank you for your nice presentation and to attend our Congress. Is there any questions about the study of Dr. Yalçın? Okay.
Thank you.
Thank you for your dear respected participants. I would like to invite the next speaker. Next speaker is Fatih Şevki Erkuş. He will present his presentation anaerobic digestion of lignocellulosic content using cattle manure and ruminal waste. Yes, we are listening, Dr. Fatih.
Thank you. Good day, everyone. I'm going to give a little introduction on our ongoing study, startup of an agricultural biogas plant using cattle manure and rumen waste to enhance anaerobic digestion of lignocellulosic content using cattle manure. We used ruminal waste as an inoculum. Lignocellulosic biomass has non-uniform three-dimensional structures consisting of cellulose, hemicellulose, and lignin. Cellulose consists of long-chain glucose molecules. Hemicellulose is an amorphous component formed by mixed C6 and C5 sugar. Lignin is a glue that provides overall rigidity to the structure of plants, includes phenolic polymers. Common lignocellulosic biomass contained 35 to 50% cellulose, 22 and 30% hemicellulose, and 10 to 20% lignin. Anaerobic digestion has been used to produce renewable energy and to benefit from the fertilizer value of the organic waste. Anaerobic digestion involves mainly 4 different steps performed by spatial microorganisms. First 3 fermentation steps performed by bacteria, hydrolysis, acidogenesis and acetogenesis. The last step of the anaerobic digestion is achieved by archaeas named methanogenesis. In order to start up of an anaerobic bioreactor efficiently, we need a good combination of anaerobic inoculum consortia. In this study, we use different mixture ratios of fresh manure and fresh rumen waste in order to enhance biomethane potential of these 2 substrates during anaerobic co-digestion. Cattle manure is mainly used as inoculum in the startup of agricultural biogas plants or as a co-substrate in the anaerobic digestion of lignocellulosic feedstock. Rumen fluid microbiota is also considered to be effective in lignocellulose digestion. However, there are limited studies in which ruminal waste is applied together with cattle manure as co-substrate. Biomethane potential is a lab-scale biological test to evaluate the potential biogas volume generated from a substrate. In this study, we used cattle manure as substrate and rumen waste as inoculum to enhance biogas production by mixing 2 or more substrates called co-digestion. First thing to do is set up a BMP set that compares mono and different mixing ratios of co-substrate. BMP test has also lots of standards, namely standard methods, OECD standard, and German method. We used German method in this set. For batch bioreactors with wet volume of 6 liters were operated in order to determine the co-substrate inoculum mixture ratios to be applied at the beginning of the bioreactor. Bioreactors were operated in anaerobic and mesophilic conditions. In order to see the inoculum effect better, buffer solution to balance the pH was not added to the bioreactor content. Cattle manure and rumen waste were diluted with distilled water to same volatile solids content, about 25 gram Vs per liter. Reactor 1 and reactor 4 were monosubstrate and the other 2 were co-substrate reactors. Substrate to inoculum ratios were 1:1 for second reactor and 1:2 ratio for the third one. The content of total solids, volatile solids, and volatile fatty acids were analyzed based on standard methods. Analysis of cellulose, hemicellulose, and lignin was conducted using ANCOM fiber analyzer. Biogas volume was measured with wet gas counter, and methane content was measured with biogas analyzer. The obtained biogas was collected daily in 5-liter gas sampling bags. fresh cow manure collected from the research and application farm of Van University. Samples of rumen waste were collected from local slaughterhouse in Van. Characteristics of cow manure and rumen waste after dilution are presented in Table 1. Table 1 shows chemical and physical characteristics of the biomasses used in this study. Methanogens are slowly growing microorganisms sensitive to low pH environment, which signals an inhibition. The acceptable pH range in anaerobic digestion is theoretically between 5.5 and 8.5. Hemicellulose and cellulose contents of both substrates were about the same. Rumen waste lignin ratio was relatively low. Figure 1A and B shows the daily specific biogas yield and methane concentrations. Low initial pH values of rumen waste, relatively cautious, long lag phase of microorganisms, but the recommended pH values were observed after lag phase for monosubstrate rumen waste bioreactor. The volume of produced per gram of volatile solids, degraded and composition of the biogas are measure of biodegradability of the material, and these two parameters also give us a raw idea of the appropriate solid retention times. The same lag phase is observed for co-substrate cattle manure and rumen waste mixtures, but the range of specific gas production rates were higher, with a range of 25 to 30 milliliter for biogas compared to monosubstrates. The obtained biogas amounts reveal that co-digestion of cattle manure and rumen waste is an effective treatment for biogas production, which provides high cumulative biogas yield with stable performance, with one-to-one application of rumen co-substrate, 60% biogas and 49% methane production efficiency increase was achieved compared to monosubstrate cattle manure. Hemicellulose and cellulose removal efficiencies were high compared to monosubstrate cattle manure bioreactor. At the end of the 25th day, when the bioreactors reached a stable state, the VFAs detected in the effluent sludge remained below the limit of 2 grams per liter and did not cause an inhibition. Results in this study further confirm that the co-substrate addition of rumen waste can enhance the lignocellulosic degradation of rumen fermentation. It's recommended to examine co-digestion of cattle manure and ruminal waste under continuously operating conditions with varying organic loading rates for future studies. Thank you for your attention.
Thank you for your nice presentation, Dr. Erkuş. If you have any questions about, dear participant, do you have any questions about the study? If you have any questions about the study, you can write directly to Dr. Erkuş. If it is possible to write your email to chat box, and they can directly communicate with you about your study and your interest about your study. Now we can go on the next presentation. I would like to invite the next next speaker for presentation. I would like to invite Krami El Mahdi, computational analysis of imaging NS SMP in human SDXBP1 gene involved in early infantile epileptic encephalopathy, molecular modeling, and dynamic study.
Okay, thank you, moderator.
Okay. Now your presentation is okay.
Did you see my share screen?
Yeah, that's okay.
Okay, thank you. Thank you. Dear audience, let me introduce myself. I'm Kramil Mehdi, a PhD student at Laboratory of Genomic and Human Genetics in Pasteur Institute of Morocco. In this presentation, I would like to discuss the result of our study entitled Computational Analysis of Damaging Non-Synonymous SNPs in human STXBP1 gene involved in early infantile epileptic encephalopathy. It's a molecular modeling and dynamic study. To introduce early infantile epileptic encephalopathy is a brain affection known as Ohtahara syndrome, which include West and Lennox-Gastaut syndrome. The affection is characterized by a preferential early age of onset, tonic seizure, and infantile spasm, leading to a deregulation of brain functions. The STXBP1 is a gene that encodes to the syntaxine 1A binding protein composed of 3 domains. The protein is expressed in the brain and is suspected to be involved in synaptic vesicle exocytosis by regulating the fusion of the synaptic vesicle with the plasma membrane. The aim of this study is to identify the most deleterious non-synonymous SNP and their pathogenic impact on the protein structure using molecular modeling and molecular dynamic simulation. To achieve the objective, we firstly collect the SNPs from DB-SNP. Secondly, the pathogenic effects of SNP were predicted using Churchy prediction tools. Then the 3D model of the wild type and its mutant form were generated using Swiss model and visualized by Yasara software. Each model has been minimized, and finally, the pathogenic effects of stability and the level of protein compaction were analyzed using dynamic simulation performed by GROMACS package. The slide presents the steps of molecular dynamic simulation. Concerning the result, the figure showed the STXBP1 protein generated using Swiss model, which is composed by 3 domains. Out of 245 non-synonymous SNPs, only 11 were predicted as deleterious by at least 9 tools. The difference between the results of the prediction software used in this study revealed the importance of using more than one algorithm to estimate the effects of on structure and function protein variation. The distribution of SNPs presented in the gene showed that the majority of SNPs are located in the intronic patch, whereas the missense variant present in the coding patch represent 1% Churchy. The conservation analysis performed using conserve showed that the 11 SNPs were located in conserved region. Indeed, several studies showed that the highly conserved amino acids are located in biological active site. And when these residues are substituted, the biological activities are affected. The 11 SNPs predicted as the margin showed a loss of hydrogenic and hydrophobic bond compared to the wild type protein. However, many studies have shown that 80% of disease related to non-synonymous SNPs produced a protein which is unstable. The analysis of the molecular dynamic simulation trajectories for 500 picoseconds was performed using RMST. The RMST analysis indicates that the variant in the picture C increased the stability of the protein, while the variance in the picture A reduces. Parameters such as overall dimensions and the compaction levels of the molecule were evaluated by RG analysis. The results showed that the variant in the picture in the middle had a highly level of compaction compared to the white type protein. To conclude, in this study, 11 non-synonymous SNPs were identified as pathogenic variants, which affect the protein structure either by the loss or the gain of hydrogen or hydrophobic interaction, or by influencing parameters such as protein stability and compaction. The analysis of the results presented here underlines the importance of all the variation involved in syntaxin 1A binding, which may be at the origin of destabilization of the complex and consequently the occurrence of the disease. The study was published in the journal Biomed Research, and thank you for your attention.
Thank you for your nice presentation about your study, Kermal Mahdi. If you have any questions about, do you have any questions about the study, the dear participants? If you have interest on this study, you can directly write to Kermal Mahdi. Please, is it possible to write your email to the chat box, and maybe if some other further questions and maybe further collaborations will be held to you directly, they can contact you directly. Okay. Now I would like to invite the next speaker for presentation. I would like to invite Sana Kantas for presentation. Presentation is about impact of climate change on ecological balance and biodiversity. Yes, Dr. Sana, you can present your presentation.
Okay. Morning, ladies and gentlemen. My name is Sana Kantas, a PhD student at University Hassan II. Today, I'm presenting one part of my research related on the impact of climate change and ecological balance and biodiversity. As a start, we will go with a brief introduction, followed by materials method, resultant in discussion, and finally conclusions. My subjects aim to raise public awareness of the impact of climate change and ecological balance, and also for studied adaptability of amphibian if this global change. Morocco is known for the richness in biodiversity ecosystem, among which we find wetland considered as one of the most important and precious richness in fauna and flora, but these environments are very sensitive and threatened by several parameters, thereby impacting the environment, biodiversity, and economic balance of the site. This ecosystem, according to data from Ramsar in Morocco, represents an area of 316,086 hectares, spread over 38 sites, including several living which we find amphibian, who consider a biological indicator of the quality of water and air. There are two main orders. Irodere represented by 2 species and 9 species of animals. These species spend most of the living in the environment which we are experiencing degradation, pollution, drought, urban invasion, appearance of new predators, example of Bibiscus ibis in region 8 million in Casablanca, and global warming. All these variations, especially thermal variation, directly or indirectly change the habitat and several of these species. Due to climate change, regeneration is one of the most affected phenomena in amphibians. These animals are able to regenerate their membrane, but for most species, this ability is limited to the large stage or tadpole. Our study aims to confirm the impact of variation of temperature on the capacity of regeneration for the hand lamp of amphibian. To understand what happened, the collect of sample had been processed in different areas, such as Diaforest Mamora, Diaforest Ben Sliman, and Surgeon from Casablanca, and over a period stretched from March to April. The tadpole collected were acclimated in laboratory conditions, and these tadpoles were kept in tank filled with dechlorinated water, 100 ml per individual, until the hand apart. The study was processed on bifomorithanic tadpole to compare the effect of different temperature on the regeneration phenomenon. The study is accomplished by different technical experiment monitoring in laboratory at different temperature, 4 degrees, 22 degrees, 38 degrees, and thermal shock. Macroscopic and microscopic observation will allow us regular monitoring on the phenomenon, and the molecular study will be performed to observe if there are appearance of new protein in case of stress. For that, we performed anesthesia of animal before starting our process. To start the experiment part, we inputted 1 part of 3 of the hand lamp of the amphibian, and that are placed in individual tank containing 100 ml of dechlorinated water, and antibiotic whole concentration is determined beforehand for each individual. For the input fragment, is immediately placed in a conserver solution containing acetic formaldehyde alcohol, lasts 24 hours before the histological study. The tadpole were subdivided in 4 groups of 8 individuals belong to the same stage of development for each phase of temperatures. And each group is subdivided in 2 subgroups, control group and average group. Group A exposed to the temperature for 4 degrees for 2 hours a day. Group B was exposed to a temperature of 38 degrees for the duration of study experiment. And Group C exposed to the thermal shock for 1 hour at 4 degrees and 1 hour at 38 degrees per day. For the last group, was exposed to the temperature of 22 degrees until the end of the experiment. For macroscopic and microscopic study, it will allow us to observe and regularly monitor the evolution of regeneration phenomenon. For the histological part, already mentioned in my previous slide, the fragments were put directly after ablation in the formaldehyde acetic alcohol. This solution composed of ethanol 80 degrees and glacial acetic acid, formaldehyde 38%, and sucrose. The fragment is emerging in the large volume of fixed liquid 10 times more than volume of pore sample, answering that doesn't adhere to the bottom or the wall of the container, and the container must be size enough to prevent deformation of the part. The duration of the fixation varies according to the volume of the sample. In our case, it must not exceed 24 hours. For second stage, test water must be replaced by paraffin, but these media are not miscible with each other. To do this, the fixed part must be dehydrated in alcohol but in increasing degree, then in xylan, and finally paraffin. The duration of the drying cycle depends on the nature of the sample and their size. In our case, we have a small fragment, so the treatment duration does not exceed 3 hours. Then we moved into the section part. The paraffin block are cut with the microton to thickness of 4 degrees, for micrometers, And then sections are collected on glass slide, followed by coloration. For this, we use hematoiosine, then move on the accessibility blade, and finally observed under the microscope and taken the picture. For the result appraisal of the test, the individual of group A development is stopped at higher stage, which was a very long compared with of the others group because of the slowing down of the mechanism due to the cold. None of this individual regenerated or metamorphosed because it did not support the cold. For group B, individual development are arrested at the hard stage. None of these individuals have originated their hand lung. The subjects are metamorphosed rapidly and therefore they have no time to continue this process. For group C, the development is stopped at the stage of cicatrization. None of these individuals regenerate their hand line. Some individuals have metamorphosed and others not. And for the last group, exposed to ambient laboratory temperatures, 22 degrees, most of them regenerate parts of their hand line. Here are some pictures of regeneration membrane. As you see, the section was regenerated after 11th day. This is some pic of some histological sections seen by microscope. And in conclusion, the outcome of this experiment showed that the variation temperatures, hypothermic or hyperthermic, had a negative influence on the regeneration of amphibian. Our way forward is to perform other tests in order to see the influence of the molecular and structural relief. And thank you so for attention.
Dear Dr. Kantas, thank you for your nice presentation.
Thank you.
If it is possible to write your email, participants can directly contact you about their questions and about interest in your study.
Okay.
Thank you for your nice presentation.
Thank you. Thank you.
Dear respected participants, I would like to invite next speaker for presentation. I would like to invite Ali Ekber for his presentation is about determination of areas to be in hypotherapy application facilities.
Thank you, Professor Söz Tutmaz. Good morning, everybody. First of all, I would like to thank my professor and participants who brought us together in this beautiful congress. My name is Ali Ekber. I teach equestrian courses as a horse trainer at Ankara Yıldırım Beyazıt University Equipment School. I have worked with horses for 27 years. I won championships and wins. Today to you, dear participants, determination of areas to be hypotherapy application facilities, I will present our work. Hypotherapy, it is an equestrian treatment method that combines the neural, sensory, and movement system of these individuals with the movements of the horse applied by expert health personnel to the clients who are called disabled and individuals.
Ali Ekber hocam, slaytlar ilerlemiyor. Bir baştan açsak ya da nasıl uygunsa. Your
Tamam hocam.
presentation stuck, the pages
Tamam.
cannot.
Tamam hocam. Hemen.
Thank you.
Hemen. Thank you hocam. Geldi mi hocam ekrana?
Şu an geldi ama tam ekran yapın. Tekrardan tekrar bakalım.
Tamam hocam, hemen yapıyorum. Geldi mi hocam?
Şu an geldi ama ilerleyip ilerlemediğinden emin değilim. Hani süreç içerisinde bir ilerletebilirseniz.
Tabii ki hocam. Şu anda
tek bir slaytta kalıyor. Tam ekran yapıp yani
hocam
normal
şu anda bilgisayarda tam ekranda ama tekrar mı durdurup yapayım?
şu an göremiyoruz. Biz sadece tek bir slayt, ikinci slaytta takılı kaldı.
Ya
Bir
da
saniye.
bilgisayarınızda tam ekran yapmayın hocam. Elle ilerletin size de uygunsa.
Evet.
Anlaşıldı hocam. Şimdi nasıl hocam?
Tamam, şimdi tamam gibi. Bu şekilde ilerleyelim.
Tamam hocam. Hippotherapy, it is equestrian treatment method that combines the neural, sensory, and movement systems of these individuals with the movements of the horses applied by expert health personnel to the clients who are called disciples and individuals. Hippotherapy practice facility. These are the areas where the clients and clients' family members, special health care team trainer, tow track site holder volunteers actively use in hippotherapy sessions and the same time hippotherapy horses lead their lives. Having adequate facilities in hippotherapy practice is important for the efficient and safe conduct of the sessions. A field study was conducted in Ankara, which is located in the central Anatolian region of Turkey, and 9 equestrian sports clubs were examined in field study. Equestrian sports clubs with the situation-based absorption method and the areas that should be in the hippotherapy application facility attention to be determined. In the equestrian sports clubs facility examined the areas in the chart were determined and sure horse welfare, hippotherapy work team and customer health and safety. The areas determined in the equestrian sport clubs facilities examined were okay. It has been observed that the facility structure for hippotherapy application couldn't be determined in the central Anatolia region, but there are equestrian club facilities, which are commercial recreation facilities that can be used when needed. According to the results of the study, the areas that should be minimum in hypotherapy eclipsation facilities, exercise room, client family monitoring area, cafeteria, horse training area, horse rest area, horse stables, horse equipment room, horse wash area, horse solarium, horse feeding room, training workroom, medical team workroom, meeting room, locker room. There should be first aid room, secondary room, cam room, toilet, shower section. For individuals and insertion, planned, attachment, hypotherapy, practice facility, the minimum areas for the health and safety of horses and people have been determined. It is planned to work characteristic of those areas determined in the index study. Thank you for listening to me.
Dear In, thank you for your presentation. If you have any further, if the presenter have further questions, you can write your email to the chat box, and they can directly communicate with you about your presentation, about your study. I would like to pass the next presentation. Next presentation will be held out by Muhammad Haris Han. His presentation is about knowledge, attitudes and practices survey regarding COVID-19 in Punjab, Pakistan.
Hello, am I audible?
Yeah, we can hear you.
Hello, good morning. First of all, I would like to thank all the moderators for providing me opportunity to present my study, and I would like to introduce myself. I am Muhammad Haris Khan, and I'm currently a student of MPhil Epidemiology and Public Health in the University of Veterinary Animal Sciences, Lahore. And the topic of my research is knowledge, attitude, and practice survey regarding COVID-19 in Punjab, Pakistan. And as we know that coronavirus have been associated with significant disease outbreak in the Middle East and East Asia over the past 20 years. And the latest epidemic has challenged Pakistan's medical, public health, and economic infrastructure in a serious way. However, based on this background, the current study was aimed that the main objective was to evaluate the knowledge, what the people are thinking about the COVID-19 and how they are handling the situations and how they are reacting to the certain limitations in terms of lockdown, as the study was conducted in the period of lockdown in Pakistan. And this was the first study that was conducted in Punjab, as limited resources were available. Moreover, due to lockdown limitations, the study was conducted as a cross-sectional online survey with the pre-tested questionnaire using a snowball sampling technique. And from the Punjab, there are two different regions. One is central Punjab and the other is the South Punjab. Therefore, we select one district from the central Punjab, namely Lahore, and three districts from the South Punjab, Bahawalnagar, Dera Rajasthan, and Muzaffargarh. However, participation in this survey was confidential and consensual with informed consent provided by all potential participants. This picture actually depicts the area that was actually selected, the study district in Punjab. The red area actually shows the area from South Punjab and the area in green from the central Punjab. However, for the statistical analysis and the program was used using R version and R Studio, and for descriptive analysis and chi-scale analysis, the package EpiTools was used. That is actually the package for the R. And to find the two associations COVID-19 victims among the respondents with the overall outcome variables. One logistic regression analysis was used to get the true cut association. However, in result, around 52.6% respondents reported awareness about COVID-19 and 64.7% individuals knew about the certain clinical symptoms like shortness of breath, while 35.3% responded conversely. Around 295 individuals from the total of 405 showed a positive attitude that the plasma therapy means the plasma rich in the antibodies is effective and treated in COVID-19, while the other respected vice versa. In terms of attitude, around 66% participants agreed that the government of Pakistan has handled the situation crisis very well. And more than 70% individuals reported good practices during this lockdown by avoiding visits to the crowded places, such that weddings, gyms, social gatherings, and shopping malls. And around 185 individuals claim to wear masks when leaving home during the lockdown situation. However, around 50% responded reported that their local hospital lacked the required facilities to treat COVID-19. As in South Punjab, we don't have enough facilities to treat the situation in terms of bed occupancy rate. Therefore, the situation might be worse in South Punjab as compared to Central Punjab. On analyzing potential risk factors, our study shows a significant association between the knowledge of the plasma therapy with the presence of COVID-19 victims in their families. Moreover, the knowledge about the quarantine and the isolation and treatment concept were also significantly associated with the presence of COVID-19 victims in their families. Overall, a moderate level of care was observed among the respondents of the both Central and South Punjab, as the global pandemic is of great concern not only on the human side but also on the animal side due to the zoonotic nature of SARS-CoV-2. Therefore, the health promotional campaigns are in need not on local level but also and globally as well to maintain a positive response. And that's all for today. If you have any questions, you have my email ID. Thank you.
Thank you for your presentation, Dr. Han. It was a nice presentation. If you want, if you can write your email to chat box, and if the participant has some questions, they can directly write you. After that, you can directly communicate about the similar studies in the future. Yeah, we can. We are going on. We are going on from the with Mohammed Shahrazdi. Previously he had a sound problem. I think he solved his problem, and we are going on with the his presentation. I would like to invite him. Genetic analysis of BMP4, GLI2, and SHH genes in sheep and goat affected with the atresia ani with candidate gene approach. Dr. Sheraz, we are listening you. If you can share your presentation.
Can you hear me?
Yeah, your voice is okay, but we cannot see your presentation.
Yeah, I am just going to share. Now it's okay. Can
yeah,
you see me? Okay. Sorry, I have some issues, sound issues. Sorry for that. I think we have to start it. This is my master's research that I have conducted last year that I am going to present it here. Title of my master's research was identification of genetic variants in SHH, GLI2, and BMP4 genes in sheep and goat affected with atresia ani with candidate gene approach. This is the list of contents that I will cover it in here in my presentation. First of all, introduction. Atresia ani is the most common congenital defect in most of the animals. Having in this issue, the animal don't have anal opening due to failure of separation of anal membrane. Ultimately, the affected animal will not be defecate. It is not limited to any specific species. It is reported in sheep, goat, dog. Most of the time, atresia is found to be associated with different type of anomalies, such as gastrointestinal vertebral defects. However, its frequency is found to be 45 to 65%. The exact etiology of atresia ani is not yet reported. However, due to its association with other anomalies and different studies reported its association with different genes, so it is reported to be a genetic disorder. Therefore, we have designed this study to find the genetic variants in SHH-GLI2 and BMP4 genes, as these genes are found to be normally involved in hind development and organ induction. Now start our material and method portion. We have just selected the animals that are diagnosed by a doctor with atresia. As I have already said that this was my master's research, so we could only collect 11 samples. Out of these, 7 were sheep, 7 were goat samples, and 4 were sheep samples. Other samples were collected from complex of University of Veterinary and Animal Sciences, Ravi campus, Putoki. Animal Genetic Lab, Department of Animal Breeding and Genetics, FAPT, Ravi campus, Putoki, UAS. 3 to 5 ml blood samples was taken from jugular vein of affected animal, and the blood was transferred into falcon having EDTA and was stored in minus 20 degree Celsius. The DNA was extracted using Grimberg and the collected DNA was quantified on 0.8% agarose gel. 11 primer pair were used in this research. All the primers were designed using Primer 3 online tool. Master mixer that was used in DCR. After the samples were sent to sequencing, and the resulted were analyzed for variants by comparing them with reference sequence that was taken from Ensemble web database. This analysis was done in BLAST function of NBI website. Results, along with genetic data, we have also collected some demographic or different demographic features, such as almost 70% goat were found affected with other anomalies, and about 25% of sheep were also anomalies. One reported that there was some type of association with the natural type of feeding mode and we have also found that 25 to 30% So there is no association in our study. Found that we found 8 single nucleotides in our selected regions of GLI2. Of 8, 6 were found in BMP4 gene and 2 in GLI2 gene, but there is no gene variant in our selected region. Out of these, 1 in BMP4 and 2 in GLI2 gene were located in coding region, and rest of them are in non-coding region. This is a novel variant that was found in exon 2 of B4 gene in goat sample. In this variant, normal C was changed with thiamine, as shown in figure. This variant also caused the change in amino acid from methionine to threonine. This is another variant in exonic region of GLI2 gene. However, at this location, you can see that there is a heterozygous condition. C and D remains in heterozygous condition. These variants are in. So we have also found the structural changes in protein, but these changes, these variants were located in disorder region, so we could not predict the protein structure change. Conclusion. This study have concluded that most of the time atresia is found to be associated with other anomalies, as a result table shows that there was almost 60 to 70% have some type of disease. Moreover, there is a higher prevalence of male sample than female, and total 8 variants. Out of them, only 3 were in coding region in BMP4 and GLI2, but there is no variant in SHH gene. Gene with atresia and I. However, a larger sample size could give an additional help to find the association between atresia and I and genes variants. This was my presentation. Thank you for your attention. If anybody have any question, you are welcome.
Thank you for your nice presentation, Dr. Shiraz. If from the participants, if you have any questions to Dr. Shiraz, you can directly write him his email. Please, Dr. Shiraz, you can write your email to the chat box, and participants then directly communicate with you for further collaborations and further questions about your study. I would like to invite the next speaker for presentation. Next speaker is Safdar Ali, and he will present his study about frequency and distribution of simple sequence repeats in retroviridia genomes. Dr. Ali, can you hear us?
Hello. Am I audible to you?
Yeah, I can hear you. Dr. Shiraz, okay. You can share your presentation.
Is it visible, my slide?
Yeah, there's no problem. You can see your slides.
Okay, thank you, sir. First of all, good morning to all of you. But in India, it is already afternoon. I would first of all, I would like to thanks BioHealth Congress for giving me this opportunity to present my work. Myself, Mohammad Ghulam Jilani, PhD scholar, under the supervisions of Dr. Safdar Ali. He is the assistant professor, clinical and applied genomics labs, Department of Biological Science, Alia University, Kolkata, India. Today I'm going to present one of the research work of PhD research work, and the title of the presentation is Frequency and Distributions of Simple Sequence Repeat in Retroviridity Genomes. This tandem repeat. Tandem repeats DNA, tRDNA, also known as satellite DNA. Satellites are tandemly repeat sequence ranging from 2 to 100 of base pair that is repeated in such a way that the repeat lies just adjacent to each other on the genome. It is localized on the centromere and telomeres of the genome or chromosome. Tandem repeats can be classified on the basis of repeat motif and the total length of the array. We can see here the small segment is known as a repeat motif and is designated by small n, and the total motif is known as capital N. It is the total length of the array. So we can classify it on the basis of this is microsatellite, mini satellite, and macrosatellite. This microsatellite is the repeat. The small n is equal to 10, and the capital N is less than 100 base pair. Then it is classified as a microsatellite. If the small n is greater than n and less than 40, and the capital N is several kilobase maybe, then it is designated as a mini satellites. And the last, the macrosatellite, is the capital N is greater than 40, and the the small n, the capital N is 250 to 500 kilobase pair, then is it designated as a microsatellite. Now, what is microsatellite? Microsatellites are made up of 1 to 6 base pair nucleotide of DNA or RNA, which is present in tandem repeated sequence in a genome. It is also known as simple sequence repeats. These microsatellites are found across the genome in viruses, prokaryotes, and eukaryote cells, and occupy both the coding and non-coding regions. The SSRs may be produced due to the error of DNA replications and repair machinery. SSRs play an important role in gene regulations, transcriptions, replications, protein functions, etc. SSRs have been used as a genetic biomarker due to its ubiquity, abundance, simplicity, variations, and multi-allelic nature. Now, we can classify the microsatellite in 6 different types. These types are pure types, interrupted pure microsatellites, compound microsatellites, interrupted compound microsatellites, complex microsatellites, and interrupted complex microsatellites. And this, in the right hand side, there is a samples of the sequence which is based on the pure interrupted compound and on this pair. Now, what is retroviridity? Retroviridity is one of the deadliest family of the viruses, causing several diseases such as cancer, leukemia, lymphoma, sarcoma, immunodeficiencies, autoimmune diseases, lower motor neuron diseases. And the family of the retroviridity have both RNA and DNA as a genetic material. RNA is a single stranded and positive sense, and the size of the genome is from 7 to 13 kbs. The variants are spirals, linears, enveloped, and 80 to 100 nanometer in diameter. The variants encoded family mainly 4 types of genes such as GAG, pro, pol, and envelope protein. It can be transmitted through several routes such as blood, saliva, sexual contact, lactating baby, and prenatal routes. It had it has wide range of hosts, including bird, monkey, rat, goat, sheep, bovine, fish, and human included. 37.7 million people infected by HIV every year. Now, this is the general applications of the microcellular satellite. If we classify it on the basis of molecular basis and structural basis, so we can see in the molecular basis, it is present. If it is present in the gene, so it is present if in the gene section, it is present in the 5 UTR, exon, intron in the 3 UTR, and it is help in the transcriptions, and it's take part in the transcriptions or translations, activate the genes, mRNA silencings, gene silencings, and transcription slippage. After all this process, it convert it express the protein or expressibility. If the protein expressed in the animals, it lead to the generations of neurodegenerative disease and cancers. If it is expressed in the microbes, it's the functions of microsatellites helps in adaptability and pathogenicity of the microbes. And if it is present in the plants, it's help in the breeding and hybridizations of the different species of plants. Now, in the another sections, if it is present in the whole genome, we see the role of microsatellite in the systematic biology. The next one is phylogenetic study, conservation biology, populations dynamics, forensic study, and parental analysis. Now we move to the methodology sections. What we have used in our lab, what is the method of? First, we used to go to the ICTV. It is a online web service where we collect all the informations of the family of the viruses. Like we collect the virus database and access number from the ICTV. After getting the access number of the from the ICTV, we used to go to the NCBI websites. Through using the access number of the particular species of the viruses, we get the total informations of the particularly viruses from the NCBI. Through GenBank data, we used to collect the informations of the detail of the viruses from one side, and after collecting the data from the GenBank, we used to get the faster format of the sequence of the viruses. Now, after taking the faster format of the viruses, we use some different types of software to analyze the sequence. First, we used to get the extractions of microsatellite from different software like MISA and IMX, through which we used to extract the microsatellites. It's a very important process through where we collect the microsatellite, SSR. We get the SSR as well as the CSSR from the MISA or IMX. After extraction of SSRs, we used another software. It is known as Science Buddies. Through these Science Buddies, we collect the GC percentage and all the percentage of the different type of nucleotides like CGA, TA. Now, we collect all the data and put it in the Excel format, Microsoft Excel. After putting in the Microsoft Excel, we do the different types of analysis like mathematical analysis. In the sections of mathematical analysis, we do the regressions part, linear correlations. After we draw the graph plot or figure plot through the Excel, as well as some different types of software which is available on the online portal like Flores Studios and DrawGraph 2. After that, we use the functional analysis for these satellites. We use different type of software like IGL-NNF and IGLSF for the SSR locations in the particularly gene. Now, we used ETE3. It is also an online free tool for the constructions of phylogenetic of the virus family. After the constructions of the phylogenetic tree, we use ITOL for the annotations and visualizations of the phylogenetic tree. Now we move to the result sections, which is in the graph portion. This is the result. Whole result is there. I will discuss about the discuss in the upcoming slide. First, I'm going to show the different type of result what we are getting. This is one of the graph which I have which is in the cursor, is showing that GC%, we use GC% as well as in the right hand side is the size comparing these, and in the below section there is a SSR and CSSR. In the right hand side we see the different parameter like SRD and SRA, and in the below section is CRD and CRA. Now I'm going to describe about the SRA and SRD. RA is a relative abundance, where the number of SSR is present in per kb of genome, whereas the RD is the total track size of the SSR in per kg per genome. And this is the CSSR, CSR 10, 20, 30, 40, and 50. I will discuss about this in the later sections. And this is the track size, what we getting, the length of the particularly SSR, whether it's the SSR of mono SSR, di SSR, tri SSR, or tetra SSR. There is a track size which we will get in our results. In the next slide, we see the different type of motif what we'll get in the mono SSR motif, di SSR motif, and tri SSR motif. The number of incidents of mono SSR is present. So highest 1570 SSR of dissections, we will get the highest SSR. Now this is the percentage of SSR in the CDS sections or in the NCS section we get. Mainly we get the SSR in the protein sections, as because of virus is made up of mainly protein. Now there is the SSR protein across the section. SSR is present across the protein. Mainly the envelope protein contain highest SSR. Envelope protein like is 511. This is the CSSR percentage present in the different genera of the family of the retrovariety. Now is the last section of the figure. This is I'm showing the phylogenetic tree of the retrovariety family. Here I'm showing the mono SSR 80 or GC percentage. In the red sections, it is a 80 percentage, and the blue one is a GC percentage. And in the there is only one black sections where I'm showing there is no 80 GC percentage of mono SSR. So here we can easily see that human related species like monkey contain mostly 80 percentage of mononucleotide, and the bird or rat contain mostly GC percentage of mononucleotide SSR. Now come to the discussion sections. Here we discussed that the GC percentage of the genome was 46%, with only one virus, RV6, belonging to the genus alpha virus, having GC percentage of 59.7%. A total of 2,732 SSR and 2016 CSSR were extracted from RV genomes with an average incidence of 39 per genome, ranging from 9 to 55. The CSSR incidence range from 0 to 10. Genome with CSSR, no CSR, there is RV 5, 7, 8, 10, and so along, with SSR range from 9 to 52. Further, the 14 genome with 3 CSSR is RV 4, 11, 14, 33, and so on, having 30 to 54 SSR respectively. Thus, the SSRs are neither conforming to any rule in the occurrence nor in the cluster across the study genome. Now come to the sections of track size. RV14-1 had the highest track size of 208 bases from dinucleotide repeats, followed by 43 RV and RV68 with 194 from trinucleotide repeats. Now RV2, RV4, RV11, 35, and 53 and 58 have the same number of 35 SSR present in their genome, but track size of from dinucleotide repeats with 156 bases, 166, 110, 112, 122, and 116 bases. The most prevalent mononucleotide motif was A, comprising of around 55% of the mononucleotide SSRs, followed by C, 167. Similarly, for di and trinucleotide repeat, the most prevalent motif was AGGA, and in the trisections GGA, CCT, respectively. All the studied genome had a minimum of 79% incidence SSR present in the coding region and the 20% in the non-coding region of the genome. The protein-specific localizations revealed almost 23% of the SSR 511 to be present in the envelope protein, with pole protein coming in the distant second position with 453 SSRs. The presence of SSR predominantly in the coding regions of RV genome illustrated two important aspects. First, since the viral genome is mostly coding, so SSR will be expectedly mostly present in protein coding area of the genome. Secondly, since the SSR associated with gene expressions and viral evolutions, their localizations in protein become a prerequisite, which is validated by the data. The increase in CSR incidence is expected with increasing Dmax and was observed herein as well. However, the rate of increase is neither uniform nor follow any single priority rule. The second aspect about the CSR worth elaborating is CSR percentage, which represent the fractions of SSR as a part of the CSR. The higher CSR would represent regions of the genome with clustering of SSR, which can therein serve as hotspots of the genome evolution. CSR percentage range from 0 to 45.71%. Thus, the uniqueness of the genome SSR signature is reiterated not in the terms of incidence and composition as discussed before, but also in the terms of localizations and clustering. And this is the last sections. Multiplaces of clustering of virus with the same or related host can be observed. On widening analysis, we can say with confidence that the contributions of mononucleotide SSRs from 80 regions is vital for host range determination. Viruses are constantly expanding their host, as in supported by HIV, which had originated in monkey, and coronavirus, which had originated bat as host. Monkey is the host for retrovirus genomes having the exclusive or near exclusive combinations of mono SSR from 80 regions. I have already discussed that monkey and human related species containing more 80% mono SSR. So lastly, the conclusion is to conclude the incidence and distributions of SSR in the retroviral genome suggested a unique genome signature, SSR signature, which is defined by the multiple factor. These include GC content evolution relations and coding and non-coding regions. We also propose the mononucleotide distributions in 80 regions of the genome as a key parameter to host divergence to human and related species. These need to be ascertained in all the known human infecting viruses. Lastly, thank you so much. This is my presentations completing.
Dear Dr. Giuliani, thank you for your nice presentation, nice and interesting presentation. If you can share your email to the chat box, the participant can directly communicate with you about your study, and maybe you can make a further collaboration for similar studies. I would like to pass the next presentation. I would like to invite the next speaker for presentation. Next presentation will be held by Gökhan Görgüşen, Gen expression analysis using Western blot analysis in Yersinia eukaryotic isolates. Yes, Dr. Görgüşen, we can see you, and if you can share your. Okay.
Good morning, everyone. Can you see my slides now?
Yeah, your slides is nice. If you can check if it is going on or not.
Okay, it's going on now?
Okay. Yeah, it's going on. There is no problem.
Okay, I hope so, yeah. First of all, before beginning my presentation, I would like to thank the organizing committee to give an opportunity to share our results. I'm Gökhan Görgüşen I'm a researcher at Van Yüzüncü University, also Valdebron Research Institute from Spain. Indeed, I'm a cancer researcher, but I contribute this research at the molecular level, and Dr. Önalan has some issues. That's why I'm going to try to explain this project. In this study, we want to determine the temperature-dependent protein gene expression level, Yersinia rickettsi, and we want to determine the gene expression profile depending on the temperature differentiation. First, we isolate the bacteria using some selective media at the same temperature, and we identified the bacteria with the morphological similarity. And then we determine their phenotyping and biochemistry properties with using the API kit, and it depends on the color changes. And then we identify the bacteria using the real-time qPCR, and depending on the 16S RNA gene, and we use specific primers to amplify these regions. When we look at our study groups, we use two different groups depending on the growth media, and then we have some subgroups depending on the different temperatures. And then for each group, we amplify these genes, 16S RNA to molecular identification of the bacteria, because our hypothesis, it just depends on the temperature differences. That's why we try to optimize all the conditions, and we try to grow all the bacteria and the equal conditions. And then we determine the gene expression profile using a Western blot, but in here we just prefer to apply SDS page and then a compatible staining to identify first we want to see the different gene expression profiles depend on the different temperatures, and then we can identify the proteins and using the specific antibodies to see the real expression differences between the proteins and depending on the temperature differences. And when we look at the findings, first we ground the strains, different media condition, and then they developed these bacteria in different temperature. And we developed some tests, and these are the gram-negative tests and the biochemical features. And we observed that Yersinia rickettsi was lost its motility and mobility at the 37 centigrade degrees. When we look at the biochemical features of the bacteria, when we see that it depends on the temperature differences, for example, the stretch uses and the gelatin production, it depends on the temperature. That's why these bacteria can lead to development of some disease. It depends on the water temperatures in the fish. I'm sorry. And after that, we want to optimize the concentration and the number of the copies of the bacteria that we use 16S RNA. And when we look at the CT values, it's almost the same CT values because their concentration is the specific ones and equal. And then we want to look at the gene expression profile, the differences between the gene expression profiles at the different temperatures. And when we look at the SDS page plot after compatible staining, for example, 110 kilodalton levels, we see that there is a significant band in these degrees, but when we look at the 15 degrees and 37 degrees, we didn't see this protein expression. So when we look at the others, for example, in here, it's almost between the 50 and 75 kilodalton weights. There is a significant increase in 37. Maybe it's because of the inducing the decreasing in motility and mobility of this bacteria, because it's significantly decreased. And when we look at the other kilodaltons, there is a significant difference between the temperatures. So as a conclusion, when we look at the conclusion, the identification of the protein's gene expression profiles is really important to determine the molecular mechanism of the disease. It depends on the humans, and the fish is the same. So you can use some new therapeutic approach against the disease. Our study was supported by Van Yüzüncü Scientific Research Unit, and thanks for your attention. If you have any question, I would like to answer your question. Thank you.
Thank you for your nice presentation. If you have any questions, dear participants, do you have any questions about the study? Okay. If there is not any questions, we can go to the next presentation. If you can, dear Gökşen, if you can share your email by chat box, participants can directly communicate with you about their study, interest about your study.
Okay.
I would like to invite the next speaker for presentation. Next presentation will be held by Mustafa Çevik. His presentation is about investigation of the effects of some phytochemicals in Yersinia rickeri and antimicrobial resistance.
Sayın Hocam, saygıdeğer hocalarım merhabalar.
Merhabalar.
Van Yüzüncü Üniversitesi Sorundere Fakültesi'nden Mustafa Çevik. Slaytım gözüküyor mu hocam?
Şu an gayet iyi. Herhangi bir sıkıntı yok.
Tamam hocam. Çalışmamızın konusu bazı fitokimyasalların Yersinia rickeri üzerine etkileri ve antimikrobiyal direnç durumlarının araştırılması. Bu çalışmada sorbus ve moringa bitki ekstratlarının su ürünleri alanında sıklıkla karşılaşılan bakteriyel hastalık etkenlerinden biri olan Yersinia rickeri üzerine etkilerinin belirlenmesi amaçlanmıştır. Çalışmada farklı zamanlarda izole edilmiş olan Yersinia rickeri izolatları kullanılmıştır. Bakteriler kanlı agar ve TSA besi yerlerinde 26 santigrat derecede 24 saat inkübe edilmiştir. Morfolojik olarak benzerlik gösteren tipik koneliler seçilerek identifikasyon işlemine geçilmiştir. Çalışma esnasında bakteriler %15 gliserol, %85 besiyeri ortamında eksi 80 santigrat derecede muhafaza edilmişlerdir. Fenotipik identifikasyon amacıyla önce bakteriler McFarland 0.5 yoğunluğa getirildi. Bakteriler mikrogen kit kuyucuklarına nükle edildi. 37 santigrat derecede 24 saat inkübe edildi. Renk değişimlerine göre, okuma tablosuna göre değerlendirildi. Çalışmada kullanılan bakterilerin moleküler identifikasyonları amacıyla önce DNA izolasyonu gerçekleştirildi. Ardından universal primerler kullanılarak PCR analizi gerçekleştirildi. PCR amplikonları dizi analizine gönderilerek sonuçlar gen bankasında karşılaştırıldı. Antibiyogram testi için 100 mikrolitre Yersinia rickeri sispansiyonu Müller-Hilton agara ekildi. Boş disklere moringa ve sorbus ekstratları 10 mikrolitre emdirildi. 37 santigrat derecede 24 saat inkübe edildi. Oluşan zol alanlarına göre okumalar yapıldı ve antibiyotikler ile kıyaslandı. Fitokimyasalların ve antibiyotiklerin Yersinia rükeri üzerine etkilerinin belirlenmesi amacıyla sıvı besi yerlerinde 10 farklı grup oluşturulmuş ve spektrofotometre ile her 2 saatte bir ölçüm yapılacak şekilde toplam 38 saatlik bir ölçüm gerçekleştirilmiştir. Ölçümün sonlandırılması maksimum yoğunluktan sonra bakteri gelişiminin yavaşlaması ile durdurulmuştur. Bakteri izolasyonları, kanlı agarda gelişen bakterilerin gerçekleştirilen mikrobiyolojik testler sonucunda gram negatif, katalaz pozitif, oksidaz negatif ve hareketli oldukları gözlenmişlerdir. Çalışmada kullanılan bakteri izolatının gerçekleştirilen biyokimyasal test sonuçlarına göre %99.85 Yersinia rükeri olduğu teyit edilmiştir. Real-time PCR analizi sonuçlarına göre çalışmada kullanılan Yersinia rükeri referans ve izolatının sigmoidal şekilde görüldüğü gibi sigmoidal eriler verdiği gözlenmiştir. Negatif kontrol olarak kullanılan master mix kontrol eşik değerinin altında olduğu şekilde görüldüğü gibi düz ve çizgiler verdiği gözlemlenmiştir. Real-time PCR analiz sonrasında elde edilen PCR aplikonları 27F 1492 reverse primerleri kullanılarak 16S ribozomal RNA gen bölgesinin dizilmesi gerçekleştirilmiştir. Elde edilen dizi sonuçları ICBH BLAST uygulamasıyla gen bankasında Yersinia rükeri oldukları teyit edilmiştir. Çalışmada kullanılan bakterilerin 16S ribozomal RNA gen dizimleri aşağıda verilmiştir. Antibiyogram test sonrasında oksitetrasiklin ve enroflaksasin antibiyotiklerinin Yersinia rükeri üzerine dirençli oldukları ancak katı besi yerinde fitokimyasal etkilerinin daha az olduğu gözlenmiştir. Çalışmada kullanılan antibiyotikler ve bitki ekstratlarının Yersinia rükeri izolatlarının sıvı besilerinin gelişimlerine olan etkilerinin incelenmesi amacıyla oluşturulan 10 grup ile her 2 saatte bir 600 nanometredeki yoğunlukları OD ile ölçülmüştür. Spektrofotometrik gelişim değerleri kullanılarak elde edilen grafik aşağıda verilmiştir. Bu sonuçlara göre moringa ve sorbus ekstratlarının sıvı besiyerlerinde etkilerinin katı besiyerlerine göre daha iyi oldukları görülmektedir. Grafikte de görüldüğü gibi TSB ve Yersinia rükeri olan besiyeri ekstrat, sıvı besiye ekstratı mavi ile gösterilmektedir. TSB sorbus, Yersinia rükeri olan lacivert gözükmektedir. TSB moringa, Yersinia rükeri olan yeşil ile gözükmektedir. TSB oksitetrasiklin ve Yersinia rükeri olan açık yeşil ile gözükmektedir. TSB enroflaksasin ve Yersinia rükeri olan turuncu renkte gözükmektedir. Diğer şekilde olduğu gibi sorbus ve moringa bitki ekstratlarına buradaki zon çaplarına baktığımızda şu sorbus, diğeri moringa, besiye yerinde bakterilerin gelişimine etkileri antibiyotiklere göre daha az olduğu gözlenmektedir. Ama sıvı besiyerlerinde bu oran şuradaki sorbusun grafikte gördüğümüz gibi sorbus ve moringa bunlar sondakilerde oksitetrasiklin ve enroflaksasin bakteri gelişiminde antibiyotikler her iki katı ve sıvı besiyerinde gelişimde büyük oranda etki etmektedir. Buradaki bizim fitokimyasallarımız ise katı besiyerinde sıvı besiyerine göre gelişimlerini %40-50 oranında önlemektedir. Bu çalışma sonucunda izolatların gram negatif, katalaz pozitif, oksidaz negatif ve hareketli ve tipik Yersinia rükeri kolonileri oluşturduklarını gözlemlenmiştir. Mikrogram ID paneliyle gerçekleştirilen biyokimyasal testler sonucunda %98 %99.85 oranında benzer olduğu belirlenmiştir. İzolatların sekans analizi sonrasında 16S ribozomal RNA gen bölgesi ile %100 örtüştüğü ve filogenetik analizde izolatların %99 benzer oldukları görülmüştür. Antibiyogram testi sonrasında oksitetrasiklin ve enroflaksasin antibiyotiklerinin Yersinia rükör üzerine dirençli oldukları ancak katı besiyerlerinde fitokimyasalların etkilerinin sıvı besiyerlerine göre daha az olduğu gözlenmiştir. Spektrofotometrik olarak TSB besiyerlerinde gerçekleştirilen ölçümler neticesinde ise fitokimyasalların %40-50 oranında bakterilerin gelişim yoğunluğunu azalttığı gözlenmiştir. Antibiyotik direncinin öneminin her geçen gün artmakta olduğu günümüzde antibiyotik kullanımına alternatif olarak ve ekonomik yönden daha uygun olduğu bitkilerin tedavide olumlu sonuçlar vereceği kanaatindeyiz. Dinlediğiniz için teşekkür ederiz.
Teşekkür ederiz. Dear Dr. Mustafa Çevik, thank you for your presentation. If you can write your email to the chat box, participants can directly write you about their studies, about interest about your study. Thank you for your participation. I would like to invite the next speaker about her presentation. I would like to invite Dr. Mahinur Durmuş İskender. Her study is about the effect of COVID-19 vaccine literacy on attitudes towards COVID-19 vaccine among university students. Yes, Dr. Mahinur, we are listening you. If you can
Thank
share
you.
your presentation. Okay, there's no problem with your presentation.
Okay. My name is Mahinur Durmuş İskender. My subject, the effect of COVID-19 vaccine literacy on attitude towards COVID-19 vaccine among university students. The World Health Organization has declared SARS-CoV-2 infection as a pandemic as of March 11, 2020, and the disease has affected the entire world. The coronavirus outbreak has caused serious health, social, and economic problems with approximately 187 million cases and 4 million deaths as of the beginning of 2020. Therefore, widespread recognition of the vaccine is considered extremely important in the fight against the pandemic. Information about vaccine tends to be complex. Understanding this information requires certain literacy and numeracy. Otherwise, the low vaccine literacy affects individual willingness to be vaccinated negatively. University students access to social platform and scientific information facilitates their research on vaccine. The COVID-19 pandemic is the most important health problem of our time, and no study has been found on vaccine literacy and attitude towards vaccine among university students, which would reveal their willingness to research and access information about vaccines developed for COVID-19. This study was planned to determine the effect of COVID-19 vaccine literacy on the attitude towards the COVID-19 vaccine among university students. Research question: is there a relationship between sociodemographic characteristics and COVID-19 vaccine literacy among university students? Is there a relationship between sociodemographic characteristics and attitude towards the COVID-19 vaccine among university students? Is there a relationship between COVID-19 vaccine literacy and attitude towards the COVID-19 vaccine among university students? Design this descriptive and cross-sectional study was carried out among university students with an online structure question between September and October 2021. Data collection tools: the data were collected using a question which consists of a demographic information form, COVID-19 vaccine literacy scale, and the attitude towards the COVID-19 vaccine scale. The data of the study were collected through an online survey link. The data collection tools were prepared through Google Forms and shared over social media groups of the students. After clicking the link of the survey, the participants were directed to a section that gave brief information about the research and confirmed their voluntary participation. After their approval, they filled in the form. Student confidence was ensured and their answers were viewed on Google Forms only via email address of the research. Data analysis statistical package for Windows version 20 package program was used to evaluate the research data. Ethics considers in order to conduct a study, writing permission was obtained from the Ethics Committee and scientific research permission from the Ministry of Health, permission from the owners of the scale used in the study were obtained. Results, it was found that the COVID-19 version literacy was low. Positive attitude towards the version were determined to be high. Furthermore, a statistical significant positive and weak level relation was determined between the negative attitude subscale point average and COVID-19 version literacy scale total point. It was concluded that the positive attitude towards the COVID-19 version decreased as the COVID-19 version literacy increased. In conclusion, it was determined that university students had high positive attitude towards the COVID-19 version, but low levels of version literacy. For this reason, students should adapt version literacy to daily life during the pandemic process, and it is recommended to provide adaptation with update information, especially in the COVID-19 pandemic where social life is limited. So social media is frequently preferred as the source of information among young individuals. Therefore, it is recommended to make strategic plans in university education about accessing the correct research as a source of scientific information about the COVID-19 pandemic. Reference. Thank you for listening to me.
Dear Dr. Minor, thank you for your nice presentation. It was the last presentation of today, this session. There is a missing one. Dr. Jamila cannot connect. There is a connection problem. As a result, we are now finishing our session. For whole presentations, do you have any questions to the speakers? If there is not any other questions, I would like to finish this session. I would like to thank you for inviting me as a moderator, Dr. Associate Professor Dr. Damla Aslan Acaröz and also the Associate Professor Ulaş Acaröz. Thank you for
Dear Söz Tutmaz, we really thank you for your support and help. I cannot find any word. Thank you for your cooperation. Thank you. Thank you for your time.
It was a very nice scientific high quality congress, and it could be in this COVID-19 status. It's very nice to collaborate and to present studies in this session. It was very nice. Thank you for all of the organizing committee of the Congress.
Thank you. So in this Hall B, our session will start by posters at 3 o'clock. So during till 3 o'clock, we will be in Hall E, the other. You can use the other link for entering that hall. Thank you. Sayın hocalarım, saat 3'e kadar burada herhangi bir sunum olmayacak. Saat 3'e kadar A salonundayız. Görüşmek üzere. Görüşmek üzere. Thank you and see you everyone. İbrahim hocam çok teşekkürler.